Good morning, everybody. Welcome to the MPG Primer. We have Raymond Walters talking about heritability of common complex traits. He is an instructor in investigation in the Analytic and Translational Genetics Unit at MGH and a group leader in the Stanley Center for Psychiatric Research here at the Broad Institute. He joined the Broad in 2014 as a postdoc with Dr. Benjamin Neal after completing his PhD in quantitative psychology at the University of Notre Dame. Dr. Walters has extensive experience in large-scale analysis of human genomic datasets, including as a lead analyst in the Psychiatric Genomics Consortium in multiple projects investigating patterns of heritability, missingness, and latent factor structures across the UK Biobank. Thank you, Raymond.
Thank you. Good morning, everyone. I'm going to dive in this morning to the fun topic of heritability, mostly in the context of common complex traits, at least the initial things we talk about generalized to kind of all traits. But gonna spend the time this morning kind of starting from just kind of as a theoretical concept, what do we mean by heritability? And then spend a fair amount of time talking about how we actually go about estimating that quantity and some results of what that looks like in practice. And then a little bit of time at the end to kind of further motivate, like even if this quantity I introduce of heritability isn't necessarily of interest to you itself, the ways in which it informs kind of other directions within genetics research. So kicking off the top on just what do we mean by heritability, kind of the very old and kind of very qualitative observation is simply that children resemble their parents. This kind of continues over generations within families. So some idea of there being inheritance of traits, and so that trait is heritable. You can inherit it from your parents. And kind of early statements about thinking about this as a more quantitative idea go back to kind of like the earliest concepts of regression and work by Galton and others, kind of just making the statement more formal that, for example, if you look at the height of a child relative to the average height of their parents, that you do see a relationship there in this kind of regression style, and then thinking about what that means for how much of that trait is inherited versus coming from other sources. And so to kind of formalize that into a mathematical quantity, at least in some intuitive sense, we can think about any given human phenotype as being some combination of a genetics piece and an environmental piece. And then the heritability from that simple model is simply saying how much of that overall variation in the phenotype comes from that genetic piece as opposed to that environmental piece. So kind of splitting up that pie chart of the kind of total variability in that observed trait. And that very simple conception that everything else we'll talk about rests on makes a lot of kind of simple intuitive sense until we have to start dealing with a lot of kind of the mathematical things that pop up out of this being an oversimplification. And so to maintain that definition of heritability, we have to make decisions about what do we do if that genetics risk for the phenotype and the environmental risk for a phenotype are correlated or if they interact. And so we could choose to take this in a direction of that genetics piece being just some sort of causal definition of the genetics. And thinking about some kind of true underlying causal effect sizes there, or we could think about this in terms of the maximal variance explained by a genetic risk prediction. And essentially the environment then becomes the residual for what could be explained by the genetics, regardless of kind of the underlying causal mechanisms there. And we'll see that pop up as we get into actual methods for trying to estimate this quantity. The other thing that emerges just from looking at this definition is that these are quantities that aren't necessarily stable across time and place, or even how you measure a given phenotype. In that if the environment changes, if suddenly there is like an increase in the variability of environmental risks towards a phenotype, that'll shift the heritability because now as a proportion of that total variability, the genetic contribution has shrunk. Similarly, if as population demographics shift over time, or you look in a different population, the variability of that genetic component may differ due to drift in allele frequencies, selection, assortative mating, all these kind of population genetics forces that will also change that heritability. And so what we have is a parameter that describes a fraction of a trait that might be attributable in some way to genetics, but that is very much going to be attached to a given population at a given time and place. And so while it's easy to fall into the idea of thinking about there being a heritability of your favorite trait, so the heritability of height is 80% or something like that, that is very much a contextual statement rather than some fixed statement about the trait. Another thing we'll run into with this definition is, and you may already be catching on to if you've seen discussion of heritability before, I've written this with a capital H, but most of the times you'll see heritability, the notation with a lowercase h, and this reflects that we have to make a decision about what all is captured in the types of genetics we're going to include in that numerator of this fraction. And especially in the GWAS era, but even before that, often we talk about heritability in a slightly narrower sense than what is this broad sense heritability. And so in particular, there's kind of a range here, starting from that broadest sense where when we say, we're going to say genetics means all of genetics, but we can quickly kind of subset that to, well, all of genetics is computationally hard, and there's some benefits to simplifying it too. We'll just talk about additive effects of genetics, because that's easier to estimate. And so that's what that lowercase h square comes from, is that narrower sense of just the additive contribution of genetics. Getting below that, and as we've gotten to being able to do genetics directly off of measured genetics as opposed to inferring it from things like family structure, we can think about the genetic contribution that is coming just from common variants and or common SNPs. And so the kind of standard notation that's emerged in the field for that is this h square sub g, just to denote that kind of common genetic piece. And so leaving aside that we're going to get to estimation methods that are using GWAS data, and so don't capture necessarily CNVs, maybe aren't capturing the contribution of rare variants, or especially de novo variants, which are kind of in this weird space of they're a genetic component, but they're not inherited. So do we want them in the definition anyway? So that kind of shift. And then if you go back to some of the early days of GWAS, there was also some discussion of the later two quantities, thinking of kind of the variance explained by a PRS or the variance explained by the top significant loci in a GWAS as kind of a heritability-like quantity, in the sense that they very directly are this kind of ratio of what's explained by that more directly measured observable genetics relative to the phenotype, but now less a theoretical quantity about kind of overall contribution of genetics and more a directly observed quantity off of some specific attempt at measuring genetic risk via PRS or from some individual loci. And so I ordered them this way in part because there is a strict ordering of these quantities. And so the heritability will range between 0 and 1 because it's a fraction of that total variability. But the narrow sense heritability has to be less than or equal to the broad sense heritability, in that either all the effects are additive or there are some additive effects, and then there's some fraction beyond that from dominance effects or other such things. That would make the broad sense be larger than the narrow sense, and that kind of continues down this chain. So those are all different versions of how to define the heritability. There's another problem that arises for many of the traits we care about, in that this kind of idea of separating the variants into a genetic piece and a non-genetic piece doesn't immediately translate to the variance of a binary phenotype, just because a binary phenotype's variance is a function of its mean and doesn't have this natural partitioning in some additive linear model kind of sense. And so the classic solution has been to imagine, as kind of already exists for things like logistic or probit regression, that there's some underlying continuum of risk that is a continuous value that we refer to as the liability for that phenotype. And then we can talk about the heritability of that continuous liability as reflecting the heritability that we care about, rather than the heritability defined directly off of the observed phenotype. And so kind of graphically, the idea here is starting in that top panel. So if we have the combination of a distribution that is the environmental effect and the distribution that is the genetic effect, we'll just get the cursor so I can actually gesture at this. Perfect. So an environmental piece and a genetic piece that combine into some population distribution that is continuous like this. And the thing that matters to the phenotype is the combination of them here. And so if we had a continuous phenotype, this might be directly the observed phenotype. But for a binary phenotype, we'll imagine that this is some unobserved underlying liability. And then so for that binary phenotype, where the statement is that that same kind of combination of environmental and genetic risk combining into kind of the observed phenotype still exists, but now what we observe instead of that smooth continuation is simply the distinction between these points in black and these points in red based on cases being above some threshold in that continuum. And so that kind of mathematical sleight of hand solves our question about how to define heritability for a binary trait. The other way you can think about this, if you prefer the kind of equation statement, is that we do the swap to that the phenotype that is genetics type plus environment is the liability, and what we observe is a function of that liability being above or below that threshold. So putting all those pieces together and trying to summarize, like, what are you talking about with heritability? So we're focused on that idea of a proportion of variation in a trait that comes from or is explained by genetics. This is, again, a population parameter in that, it can shift based on how we measure the phenotype and who we measured it in and the forces happening in that population. And kind of depending on our decisions about how exactly to define that genetic piece versus that environment piece, it might make sense to think about this as like a causal proportion of the phenotype, but it may also make more sense to just think about it as the most you could explain by the best possible genetics predictor, possibly subsetting to, say, common variants or, observed variants in some way, but is kind of the population level observed underlying like best possible prediction level. What that means for what it is not is that it's not a statement about how mutable the phenotype would be over possible interventions or amount of variability that could exist in shifts in the environment. There's a lot of good literature trying to hash out kind of the theoretical statements of this, especially out of the sociology literature and especially around debates over the heritability of things like intelligence. And perhaps the cleanest summary I've heard goes back to this paper from Goldberger, who remarks that we could find that the heritability of, say, eyesight maybe approaches 100%. Practice, it probably doesn't, but for sake of argument, we could say it does. But that wouldn't mean that we necessarily need to give up on the idea of making glasses for people. Like we can, just because a biological phenomena has some contribution of genetics and some variability in the population doesn't really speak to the opportunity to intervene on it. It is simply a measure of the variability as it exists at the moment. The other thing is because it's a population parameter, it doesn't tell you anything really about the difference between populations. Like other than that, you could say that the heritability in some population for a phenotype is higher or lower than in a different population for that same phenotype. That doesn't tell you about whether observed phenotypic differences between those populations are due to genetics or not, because it's a within population parameter rather than a between population parameter. So all those are statements about kind of heritability as a theoretical quantity. And now we can get into, in practice, how do we actually go about trying to estimate this quantity? And that estimation historically started in the same place the observation about what we even mean by this concept of heritability did, which is with that resemblance within families. And so in particular, the kind of quantitative way to get there, so keeping our same model that we're talking about, the phenotype that has genetics and environment in it, if we have a pair of people, the similarity in their phenotype should reflect the combination of how similar their genetic risk for that phenotype is and how similar their environmental contribution to that phenotype is. Mathematical arrangement gives you that the heritability lives as coefficients on kind of those pieces in assembling a total correlation. And for related pairs of individuals, we don't actually need to observe their genetics directly to know, at least in expectation, how similar their genetics are, simply as by virtue of their relatedness. This is a slightly easier statement to make for like common complex traits, where we expect that the kind of overall relatedness for a pair of individuals should fairly directly translate to how similar they are on the genetic component for any given phenotype, just because polygenicity exists. It gets the level of certainty of there gets much less certain once you're talking Mendelian phenotypes, where like the level of expectation is the same, but the amount of variability in between individuals with the same level of relatedness, how often they'll have that kind of same amount of sharing will be, yeah.
Assumption here is just that this is all multivariate Gaussian, right? That goes into this. So this is just like a large number of locus assumption.
Yeah, for that same about why works better in complex traits. That's the assumption. Yeah, the one that it's a large number of loci, that kind of central limit theorem, kind of many loci contribution gets you there. You don't have to make that assumption because even in Mendelian inheritance, in expectation, that correlation works out the same. It's just that you don't have the kind of nice law of large numbers kind of behavior for getting there. Cool. So with that kind of intuition, the kind of canonical example of how this has been gone about is thinking about this within families or especially within twins. And so the idea here is that kind of schematically, if you think about pairs of individuals based on the nature of their relationship, this is kind of drawn out along the x-axis of this diagram here. Kind of the more related you are, especially for twin pairs, if a trait is heritable, you should see that that phenotype is more similar between those pairs of individuals, versus if a phenotype isn't heritable, then kind of regardless of how similar the individuals are genetically, the trait's only being driven by environmental features, and so you'll just have some kind of average background of similarity in the phenotype that's essentially null. And so you could do this with the full spectrum of relatedness, but turns out you can get there fairly easily just with twin pairs that are either identical twins or fraternal or dizygotic twins. And so I won't belabor stepping through all the math, but essentially the idea here is that the identical twins will share all of their genetics. The dizygotic twins will share half of their genetics on average, as any siblings would. We'll make an assumption that the similarity in the environment between a pair of twins is the same, regardless of whether they're identical twins or they're dizygotic twins. It's a rather questionable assumption, but is the common assumption that gets us to the easiest estimator. And so in that scenario, the heritability emerges as simply the difference, as a function of the difference in the correlation between pairs of twins that are monozygotic versus the ones that are dizygotic. Because you have that difference in how genetically similar they are, we can subtract out away that kind of equivalent environmental piece and get to just an estimate of the heritability. And so kind of as an old, simple version of getting there, you get an estimate like this. There's much room to generalize upon that model and get more sophisticated. So often this will get represented in terms of something like the ACE model, where we now have an additive genetic component, a dominance genetic component, a shared common environment piece, and a unique environment or residual error term for each individual phenotype. And we can set expectations about how correlated between the individuals each of those pieces should be. So the dominance piece will behave different than an additive piece. There's room to allow variability on what that shared environment looks like, especially as you incorporate dizygotic twins versus other forms of siblings versus cousins. You can extend this with individuals from those other forms of relatedness. And essentially get to more sophisticated estimation off of that same basic familial form of thinking about heritability. All that is done without observing the genetic data, though. And so we now have many methods that work with data that we can observe genotype and attack this question of how much genetic contribution to phenotype there is in other ways. This list isn't exhaustive, but is intended to just give you a flavor of is kind of the diversity of methods that exist. There's a wide range. And so for today, I'm going to talk mostly just about these two that are probably the most common in circulation and kind of provide a good reference point for many of the other methods here. But what I'm talking about by no means is exhaustive for the different ways that have been proposed for estimating heritability. So starting with this method called Gremmel, which comes from it's a restricted maximum likelihood estimate using genetic relatedness. And so it emerges from that exact same idea that existed for our twin or family-based estimation, that more related individuals will be more phenotypically similar, proportional to how heritable the phenotype is. But says instead of working at the extreme of like perfectly related twins, can we do that with nominally unrelated individuals based on the kind of much more subtle variation in people who are slightly more genetically similar than average or slightly less genetically similar than average? And when you play that idea out, the model that you kind of get to is that you essentially say that that variation across individuals in the phenotype and the similarity or dissimilarity between those individuals is a function of kind of a residual error term that is I, that is independent across those unrelated individuals. And then this genetic component that is proportional to the observed genetic relatedness between those people based on a genetic relatedness matrix estimated from the estimates of IBD and genetic sharing. And so well, what the literal estimation of that via maximum likelihood isn't quite the regression shown here. Conceptually, they end up in a very similar place, and that you have this kind of narrow range of very small genetic relatedness estimates that are slightly above or below 0, and a relationship to that for any given pair of individuals to kind of how similar or dissimilar their phenotype are on some standardized scale. And the very, very subtle slope that does exist in that line would give you an estimate of heritability. And conceptually, that is approximately what you're getting out of the actual way that GCDA does this in a maximum likelihood sense for fitting that model. And so kind of the key here is as long as with your observed genetics, you can make that estimate of how genetically similar people are and kind of cross your fingers in the same way we already discussed that, especially for things that are polygenic on average, that, observed amount of little bit of higher or lower genetic relatedness will track with the phenotype, because somewhere in that estimate of the genetic relatedness is the relatedness on the pieces of the genetics that actually inform the trait. And so this gives you an estimate off of that observed genetics of the heritability. Yeah.
On the left, sorry, on the left, that's the variance covariance matrix in the phenotypes, or the
in the equation, yeah.
And the I, that's not the identity matrix, or it is the identity matrix, or just matrix of ones?
That is the identity matrix. So it turns into a slightly weird statement. So the implied matrix on the left-hand side is n by n, so individual by individual. In practice, the kind of sufficient statistic observation there ends up being just the cross product of the two phenotypes between the two individuals with some standardization for scale. And so, yeah, then you have a term that's on that matrix of correlation in genetics, and in term of what is meant to be correlation in environmental terms that we take to be the identity matrix, and say that their environments are independent since they're unrelated. Cool. The other method I want to spend some time talking about is called LD score regression, which takes a slightly different tactic, particularly living within our GWAS era, and asks, so we have GWASs looking at the amount of association of all of our common genetic variants with a given trait. Here, the example of the 2014 paper on schizophrenia. And so instead of looking directly from how related individuals are, can we take our observation of the amount of association signal in a GWAS and turn that into an estimate of heritability, partially for the benefits of being able to do this, hopefully off of just the GWAS results without needing to refer back to the individual level genotypes in a way that allows us to keep up with, say, meta-analyses. And maybe some lower computational overhead. And so the intuition that turned into LD score regression was that if we imagine our genome, here a toy genome with 20 little SNPs on it, and we know that the genome has this structure in terms of blocks of linkage disequilibrium. And we'll lay down some blocks of that on our little toy example here. And we have some SNPs that aren't in LD with any of those blocks. We'll call them lonely SNPs. They have no friends. And for sake of illustration, we'll say that the SNPs in the LD blocks are all perfectly correlated with one another, no variability in that R-square, just perfect LD blocks. And so we ask the question of, so if this is our toy little genome, and if polygenicity exists and there's some set of causal variants that kind of exist at random across our miniature genome, what might we observe in GWAS association? And so if we play that out, we can say that, well, we had, there's a couple causal SNPs. We'll say at random, they fall in these two locations. And so for our GWAS, what we'd observe is that for each of those causal SNPs, all of the SNPs in that LD block would show association in the GWAS. We can't really differentiate which is a causal variant because they're all perfectly correlated. And so each of those blocks would be fully associated. The things not in those blocks would not be associated. But we're doing this kind of at random. So, maybe in a different reality, it'd look like this, or maybe in a different equivalently structured section of the genome, you see causal SNPs in these locations. And so again, you'd see their corresponding LD blocks show association. You wouldn't see association elsewhere. We play this out as kind of a random process that continues to happen. Every once in a while, we do actually hit one of those lonely SNPs. And so it doesn't show association for a block. We see association just for that SNP. Hooray. But if we thought about that process kind of happening at random at large scale, what we'd observe is that the likelihood that a given SNP shows up as associated with our phenotype in a GWAS sense would be proportional to the size of the LD block it lives in, or how much genetic variation it's tagging, kind of equivalently there. And so it turns out that that observation, with some simplifying assumptions, lets you derive a mathematical expectation that the result in the GWAS, represented here kind of in the expectation of its chi-square statistic for a given SNP, is a function of some pieces that have to do with confounding or population stratification, but then a piece that is related to that concept we just laid out, which is how much variation does that SNP tag. So in practice, taken as the sum of correlation of that SNP J with all surrounding SNPs. And it turns out that when you walk through that kind of expectation, that you have a piece that is constant with respect to SNP, because it is more about overall inflation that's not a in the GWAS that isn't related to LD. And then something that we, if we think of this as a regression equation, would be the slope attached to that information about LD. And that slope happens to be a function of heritability and sample size and number of SNPs, which are known quantities. And so we can do this regression, get a slope, and turn that back into an estimate of SNP heritability. And since the only inputs here are GWAS information and information about LD, which we could pull from some reference panel without needing to go back into individual level data, we can do this just from the information that's in the GWAS. And it turns out that we can get this estimator because there is a link between that amount of signal in the GWAS and the and that overall heritability under some expectations. Doing good at time. All right. So that's at least kind of the main ways in which heritability estimation has happened in the field. What do these different approaches tell us about heritability in our complex traits? And so kind of, especially out of the long history we have now of twin family studies, the kind of broad takeaway has been the statement that's, been described as the first law of behavior genetics, though could probably also apply to kind of human genetics writ large, which is that at least as a first approximation, all traits are heritable. Was kind of the rule of thumb that people expected, and kind of reviews of the literature have upheld that idea. So this is a review done about 10 years ago now, looking across about 50 years worth of twin studies and meta-analyzing as much as possible within traits that have been studied over that time. And found that kind of broadly across phenotypic domains and these different clusters of parts here that you see kind of this moderate heritability, often, either side of 0.5. It's in the purple bars here versus there's some information on the kind of shared environment estimates in those green bars. So kind of, yeah.
I had a question. Earlier you mentioned heritability as a population level parameter. Have people made similar plots for this for other populations, or should we assume this is the white European estimates we're looking at?
Yeah, it's a good question. So this is, the estimates in here include estimates from multiple populations going into these averages, but the preponderance of those studies, and so the preponderance of the weight in these values are coming from kind of white European predominant populations. The values do tend to be similar, up to variations that are more study-specific factors, but there's not like broad differences, I would say, in terms of the takeaways for different like ancestral populations. So in these different phenotypic domains, you see maybe some variability where some of the more anthropometric or more directly measured kind of phenotypes are maybe slightly higher in heritability and slightly lower for, personality or social values, the kind of phenotypes. But by and large, strong heritability in basically every type of measure we care about in people. And the same broad statement probably applies to our estimates off of the genetic data. So here, looking at methods with that estimates from that LD score regression method applied to GWAS in thousands of traits in UK Biobank. The particular results here are from an effort we did in the lab. But this has been kind of replicated in kind of multiple publications of different domains of phenotypes as well. And so kind of broad strokes, you can see the distribution there and the significance on those values stratified by kind of our level of certainty in those phenotypes due to sample size and measurement scales and things like that. But kind of broad statement, the middle of that distribution is solidly away from zero with an average heritability of around 0.1 in this form of estimation, looking at it. This is a SNP heritability as opposed to a family-based heritability estimate that's in theory at least all of the narrow sense heritability. But at least for all the things that are well-powered for GWAS and subject to some kind of QC, we see that the preponderance of things that are well-powered show signs of genetic signal that align with having at least some heritability out of our GWAS results. So at least kind of qualitatively concordant with the idea that everything is heritable. That difference, though, between that 0.1 that exists in like an LD square regression or other efforts that have been made to quantify the genetic signal in GWAS versus the like 50% heritability in a lot of the historic twin family studies, did at least for a time cause the kind of crisis of faith of thinking about why are these estimates in GWAS estimates so much lower than what we observed in twin family studies, the so-called missing heritability problem. And part of that goes back to kind of thinking more carefully about the different quantities that are being estimated, so that twin and family methods are really after all of narrow sense heritability, versus GCTA or LD score are at least somewhat attached to the observed genetics, and so are more in that SNP heritability, H squared G kind of estimation. And some of the statements about missing heritability were more concerned about differences down to the more PRS or top loci type variance explained estimates. But there are probably other reasons that due to differences in methods that are more than just that they're estimating different quantities. So even between LD score and granular GCTA, we observe kind of some variability where often the LD score type estimates a little bit lower. Part of this goes back to more subtle differences in the quantity they're estimating. But also there's a lot of impactful decisions that exist there about kind of implementation details, particularly around kind of treatment of allele frequency and how that contributes to what is the presumed model of signal in a GWAS, and how that contributes as far as relative weight that is captured in a genomic relatedness matrix. And so you can see wide variability, in simulation studies from different versions of these methods that attack these questions in different ways and relative to different underlying genetic architectures and distributions of where the causal effects live and things like that. And so there is room in all of these methods for different biases to kind of percolate through. And so not only be estimating different quantities, but also be estimating and making different assumptions about how to get there that could have biasing effects in either direction. And this isn't a statement just about like LD score and GCTA. This is just back in the twin and family studies as well, because we had to make that assumption about the similarity of the environment between the identical twins and the dizygotic twins. And so if that violation is assumption is violated, we'll similarly have biases there where that might contribute to those higher heritability estimates as well. Yeah.
So estimates from GWAS data, is that purely additive?
Everything I've tried to summarize here, yes. There are extensions of both the GCTA and LD score that also get into dominance effects. By and large, kind of searches across traits with those have found very little sign of dominance effects in trying to do a dominance GWAS. Mostly things that tend to be at loci that are already well captured by additive effects, and you're just looking at the residual around that's dominance. But that is also in the allele frequencies represented in GWAS, as opposed to kind of more rare variant studies where we might expect a lot of more dominant or recessive acting variants to live. Any other questions or paused? All right. Cool. So before I move on from the topic of how we do estimation, just like a quick highlight of some of the ways that these things go wrong in their respective estimation. There's kind of a garbage in, garbage out phenomenon that just because you get to like rely on somebody else's GWAS doesn't mean you aren't now attached to the decisions they made in terms of where measurement error may have affected their phenotype, where the ascertainment kind of aspects of the study design contribute in. Ancestry and stratification are real concerns for all of these methods in terms of needing to differentiate, whether they're ever going to try to differentiate ancestry-related signal from overall genetic signal, and if so, how they're going to accomplish that, because genetic similarity between individuals will include the similarity of their ancestry unless you do something to try and portion that out. But how successfully that's done may vary. We've already talked a bit about the assumptions that exist around kind of the environmental piece, whether that's the shared environment in related individuals or kind of just treating it as a residual term that we assume to be independent in the GCTA type model. And then some of the kind of tricky aspects that exist around things like actually applying a correction for getting to that liability scale amount of signal off of having an observed binary phenotype. It's easy to state what we want and as a theoretical concept generally, but actually getting that in an unbiased manner can be difficult. There's some standard transformations that work approximately fairly well, but how well they work depends a bit on just how common the phenotype is and some of the other features there. Cool. So the last bit to talk about is kind of where heritability gets into other related kind of areas of genetics research. And so without having infinite time to talk about any of these in great detail, kind of a few places of interest that I thought were worth mentioning, starting with polygenic risk scores. And so when I introduced early on, I made the note that kind of heritability, or particularly at least a SNP heritability, kind of exists as a theoretical quantity that would be the variance explained by our best possible genetic predictor, including all of those kinds of variants. And so, for the sake of thinking about polygenic risk scores, this exists as kind of in the infinite data limit. We might expect that we could reach that level of predictive power, never more than that. And so how that informs on where we think a polygenic score may or may not end up being able to be useful, or how much it'll contribute, at least has this kind of theoretical bound on it provided by heritability. In a more direct sense, even before you get to that infinite limit, you can derive expectations for how well a PRS should predict as a function of heritability and the available sample size for the GWAS that is used to create that PRS. Which generally comes down to a function of heritability, as well as so this rho G term has to do with the correlation between the discovery sample that was used for GWAS and the target sample where you're actually applying the PRS. So if there's some difference, either that you're predicting into a different phenotype, or just there are differences in that phenotype that exists between cohorts, that'll attenuate your prediction. And then as a function of kind of the sample size and number of SNPs contributing to that PRS. So you can get actually the expectations about kind of future forecasting of how well a PRS should be behaving that come out of heritability. And even in the construction of polygenic risk scores, a number of the kind of polygenic risk scoring methods now kind of incorporate heritability as part of setting priors for expectations about the distribution of effect sizes to more robustly estimate the effect sizes that should be used as the coefficients for constructing a polygenic score. So the example equation here comes from LD Pred that I won't belabor the details on, but are basically getting expectations about those effect sizes that depend again on SNP heritability. Another place where we see heritability pop up as kind of conceptually related is thinking about enrichment testing for various enrichments of signal across the genome. And this kind of follows from the idea that our null hypothesis for most enrichment testing is some idea that the genetic effects that exist for a given phenotype are kind of uniformly distributed across the genome. And we're looking for places where that doesn't actually follow and that we have some enrichment in some place. And so one way we can formalize that and get to the quantities of estimation is if we can estimate heritability in some subset of the genome, whether that's separate regions or SNPs that have some annotation or some concept like that, and get heritability that is specific to that category, these C's here, and relative to kind of the size of that annotation or how many SNPs are present in that. That relative signal in that category to the overall heritability in kind of a uniform expectation across all SNPs gets a natural statement as far as what we mean by this quantity of enrichment. And that enables all the things we see in terms of talking about enrichment in signal and GWAS studies. So things like looking at enrichment of signal in conserved regions or in regions that are associated with expression or open chromatin in particular cell or tissue types. And all of those kind of secondary analyses of GWAS that try and pull out this kind of signal relative to a baseline measure of how much overall signal there is that is showing up in different places. Heritability is also stepping stone towards other kind of genetic architecture parameters that kind of live as extensions of that same kind of model of a phenotype is simply genetics plus environment. And so one common direction to that is genetic correlation, where we say now we have that same model, but it exists for two different phenotypes. And then our genetic correlation is defined kind of by that correlation between the genetic component of trait 1 and the genetic component of trait 2. And to estimate that, it often occurs that we'll have some estimator for the covariance of those two terms and standardize that relative to the heritability of the two phenotypes. And that again gets us to our genetic correlation. So kind of extending from that one phenotype, that one phenotype variance parameter now to a covariance parameter, and getting these statements about correlation across phenotypes, extending that idea. And so, wrong direction. There you go. And so again, this has been done increasingly across phenotypes and observing kind of pervasive genetic correlations in various domains. And it turns out that like even beyond genetic correlation, other kind of moments of that distribution of effect sizes that live kind of in that same space of ideas are also informative about aspects of genetic architecture. So this pair of papers from Luke O'Connor and Alcus Price kind of get into how some of these quantities also manifest in asking questions about polygenicity or about causal relationships, again, coming from these same kind of extensions of parameters of architecture. And then lastly, in thinking about kind of selection or population genetic forces, heritability also lives in expectations about the kind of relationship between strength of selection on a phenotype and how much impact that'll actually have in shifting genetic drift over time, and how much the phenotype will actually be able to move in response to the selective pressure on the phenotype. So kind of these fundamental quantities just connect in many ways kind of across our landscape of genetic research. So just to kind of wrap up again, the kind of rundown here. So again, heritability is conceptually this proportion of phenotypic variance that can be attributed to genetics in some form or another, but is very much a population parameter and not a statement about our ability to intervene. We have a multitude of methods for estimating this, from those old twin and family studies into methods using GWAS data that each come with kind of their various hiccups and things to be concerned about in how we estimate them. But by and large, the takeaways have been that we observe lots of heritability across the human phenome. And this is informative both for those estimates directly and in the ways those estimates inform everything else. And so the slides will be distributed and also has an appendix full of kind of related resources and publications. But hopefully that it's covered the space as far as getting introduced to the idea here.
We are almost, well, we're at time. If anyone has a question, they can come up to the mic. I do have one from online from anonymous attendee. There's a question about are there any problems if we are comparing heritability across populations?
Yeah, so for comparing heritability across populations, conceptually it's possible, but often the thing that emerges is that matching all of the features of the studies to actually make that an apples to apples comparison can be quite difficult. And so, like I mentioned that even things like measurement error will affect this, and that is that like if you have measured a phenotype comparatively worse and just have more measurement error in there, that'll lower the heritability as an overall proportion of that variance. And so by no statement about how much genetics actually contributes to some like true trait there, you'll observe differences. And so getting those kind of features comparable is quite difficult. So even while in principle, that kind of comparison should be possible, in practice, it's quite difficult.
Thanks for a beautifully clear talk. Early on, you mentioned that there's this fundamental ambiguity of interpretation between kind of like direct causal, maybe mechanistic style effects versus ability to predict. And I just wanted you to say more about your views on that, if you view that as something that the field needs to grapple with and help disambiguate by, I don't know, introducing more concepts, or if it's something we should live with, and if that's dangerous, etc. Thanks.
Yeah, it's a good question. I think there's a place in the world for both versions of thinking about that estimation. And most of the challenges arise in the fact that we are applying the same name of heritability to mean possibly either of those concepts, and thus implementing methods that attack it in different ways. The version that is best possible prediction is certainly easier to get at, because then you can think of the environment as solely the residual piece. And it also gets you out of some of the problems that also exist in GWAS about, whether it be stratification or effects that are the environment that comes from your parents, and so is correlated with the genetics, but has nothing to do with how the actual inherited genetics are acting, that will show up in GWAS signal unless you're doing within family study. All those pieces are much harder to practically deal with, but there's, there is still value to wanting to think about the quantity that is the true underlying causal effect of the genetics, and having a name for that and thinking about it in concept. It's just a practical difficulty in terms of making sure that we are thinking clearly about what we've estimated and what that actually says versus what we might wish we could get to, and thinking about where the pitfalls are if what we want to think about is the causal effect version.